Hello everyone. In this presentation I'm going to show some of the preliminary results that we obtain in the framework of a numerical investigation aimed at developing an innovative diagnostic technique to detect atherosclerotic plaques in the human blood circulation. As we know, atherosclerosis is a very common and deadly cardiovascular disease in Western countries, and its early diagnosis could save lives because typically symptoms manifest too late. This work is aimed at improving the effectiveness of the diagnostic technique that I'm going to show you in a moment, to describe in a moment. And it's also in line with the trend of using direct numerical simulation and simulation of this kind in general to customize diagnosis and use simulation for virtual medical purposes. The diagnostic technique consists in a combination of two techniques actually. First of all, gold nanoparticles are delivered by injection in the blood circulation. Each golden nanoparticle is engineered by mounting pieces of antibodies at each particle surface, and this antibody will stick onto its own antigen, which is actually produced by atherosclerotic plaques. So the golden nanoparticle will stick onto atherosclerotic plaques because of this antibody. And the second ingredient of the technique, by photoacoustic imaging, it's possible to compute the concentration of golden nanoparticles and it detects the position of the potential atherosclerotic plaques much earlier than the plaque will form completely. These golden nanoparticles are small enough, 20 nanometers as you see, not to be recognized by the antibody system. For a moment, most of experimentation is made in vitro and in mice. So for this reason, we will consider both murine and human blood circulation. In particular, we will focus on the aortic arch, which is critical for atherosclerosis in mice, even though it's not critical for humans. We have used, of course, OpenFOAM to carry out the simulation. They are quite fast simulation. They took a couple of days each. And we have used the pimple scheme. First of all, we have checked, we have verified the second order accuracy, considering a problem which looks like the problem that we were going to investigate. We perform a direct numerical simulation of a pulsating flow in a straight circular pipe with rigid walls and the boundary condition at the inlet with a uniform velocity. For this problem, an analytical solution exists, which we have exploited to verify the accuracy of the method. This problem is characterized by three dimensionless numbers. For example, the von Moersley number, the Reynolds number of the tube, and the Reynolds number of the boundary layer, the Stokes boundary layer. If the Reynolds number of the tube is much larger than the Reynolds number of the boundary layer, we can expect a steady flow-like regime. Otherwise, you can go to the limit of an oscillatory boundary layer. As you can see in this figure, in the pink lines and the dots, you see that the numerical simulation approach very closely both the analytical solution and experimental measurements. And it was possible by changing the grid size, the mesh size, to evaluate also the accuracy of the method, which looks like second order, as you can see in this diagram. So Then we have assumed that blood is a Newtonian fluid, which is a good approximation for arteries, with a viscosity which is about 4 times larger than water. We have considered particles since they are so small, like point-wise, so we have used Lagrangian point particle approach to compute the trajectory of individual golden nanoparticles. As you can see in this figure, the aortic arch has a somewhat a complex shape. We have approximated this the aortic arch with a tube of constant cross-section and the rigid walls. This is an assumption, of course. And of course, we have to introduce another parameter, which is the curvature, or alternatively, the Dean number. This is actually the geometry of the simulation, with the inlet and the outlet. At the inlet, we have considered the flow rate for human on the left hand side and for mouse on the right hand side, and you can see the difference of geometries from human to mouse case in the left picture. Looks much simpler in the mouse case, but geometry is really similar, and actually it's just 20 times smaller in the mouse case. We have compared the values of the shear stress, wall shear stress, with measurements in the human case. There are many measurements for the human case, and you can see that in the left panels here, and with other simulations that were carried out with a similar geometry on the right hand side. And the comparison is qualitatively and quantitatively nice. And in the table, at the bottom, you can see the values of the dimensionless parameter that we consider for the two simulations that we carried out. Consider the flow is mostly laminar. Actually, it's laminar in the mouse case. In the human case, you can see that there are vertical structures identified by lambda 2, which are associated with the separation of the boundary layer where the curvature is too large. This separation typically occurs during the deceleration phases because the pressure gradient is opposite, indirect in the opposite direction. This is quite known. So the flow is indeed different between the mouse and human case, even though many experiments, a lot of comparisons exist between the mouse and human case, and so mouse case is used to investigate the effectiveness of techniques for humans, even though the flow is somewhat different. We should take into account of this difference. The presence of vortices is actually a good thing in the human case, because vortices means that the dispersion of golden nanoparticles will be more effective, and there will be higher probability for golden nanoparticles to reach the site where atherosclerotic plaques could form. You see that shear stress, the wall shear stress, is much larger in the mouse case than in human case. Indeed, this is the main cause why the aortic arch is critical for a mouse, because large shear stresses are strongly correlated to the formation of atherosclerotic plaques. This is mainly because platelets will start to form aggregates in presence of large shear stresses. We have computed the trajectory of the golden nanoparticles in order to understand whether their dispersion is efficient enough. You can see that the motion is somewhat different between mouse and human case. In the mouse case, their particle trajectory is mostly parallel one to each other. And in the human case, actually, it takes more time for a particle to reach the outlet, and during this time, the dispersion is evident. To evaluate this effectiveness quantitatively, we computed the correlation. First of all, we divided the surface of the aortic arch into tails by tessellation. And for each of these triangular tails, we considered a little volume close to the wall surface. And we counted the number of golden nanoparticles that enter this little volume at each phase of the pulsation. Then we computed the fluctuations of the number of these particles and of the shear stress, which we found, the wall shear stress, which we found is a parameter to quantify the risk of formation of atherosclerotic plaques. The fluctuations about the average value averaged over the surface at each phase. We can expect that where this correlation is bad, so you have large stresses and a small value of a small fluctuation, these phases are more critical for the diagnostic technique, because you have a large probability of formation of plaque and a small probability that you have a particle that reaches the surface. You can see that there is a phase during the acceleration phase where in the mouse there is a very large correlation, and this very large correlation is due to the presence of secondary motions and eventually vertical structures, not turbulent ones, which improve the dispersion of golden nanoparticles. In the human case, this is not a problem, so the correlation is always somewhat large. In order to identify exactly the existence of cases where you have a strong fluctuation of shear stress and a small number of particles, we computed a sort of quadrant analysis. And actually, the fourth quadrant, where you have large fluctuation of shear stress and a small value of the golden nanoparticle reaches the surface, this is the most critical, and you see that it is about the 25%, between 28 in the mouse and just 22 in a human case. In other cases, there is a good probability that the particle reaches the surface, and actually we didn't find any tail, so any place of the surface where throughout the pulsation cycle there were no particles. So concluding, we could say that the diagnostic technique is somewhat effective because all the parts of the surface were reached by golden nanoparticles. Even though there is it could be improved, the technique anyway, because you inject a lot of particles which cost a lot, and many of them are just lost during in the blood circulation. So if there was the possibility to make the technique more selective, that could be, of course, appreciated. In order to make the simulation more realistic, we will consider a more realistic geometry in the next future. For example, considering CT segmentation measurements, so a realistic wall, possibly elastic wall, because in the reality the surface is indeed elastic. And it will be also possible to compare the results with in vivo observation. In vivo, I mean results from mice, which is what the group is currently doing. So thank you for your attention.